Contrasting gene expression between developing honeybee, queens, and workers. Introduction. Honeybees are polyphenic species, which is shown in caste development. Polyphenism is where two or more distinct phenotypes are produced from a single genotype, usually from environmental factors. There is no genetic difference that distinguishes queens and workers. Instead, the differentiation is determined epigenetically. For example, the phenotypes show up in workers as not being able to reproduce. In contrast, queens have hypertrophied reproductive systems and capabilities. The honeybee larval diet changes gene expression, which causes female honeybees to become a worker or queen. With research, you can dive deep into learning exactly what proteins and genes are expressed in queens versus worker honeybees. Evans and Wheeler study. As you can see by the audio-radiographs, 7 differentially expressed loci are shown. You can tell which genes were more expressed in workers versus queens. The darker the blot, the more expressed the locus is. Queen larvae were more intensely expressed in loci 1CB6 and 2QA4, whereas loci 1BH4, 52A9, 52A11, and 52C8 were expressed only by workers. Locus 1CG5 was more strongly expressed in workers as well. The results prove that distinct activation of genes in queens and workers are involved in caste determination. Implications for caste-selective aldehyde and fatty acid metabolism. This study focused on identifying proteins required in the caste-selective mandibular glands. ALDH1, ALDH3, ALDH6, and ALDH7 showed significantly higher expression in queen mandibular glands than in worker mandibular glands. As you can see with the graph, queens are represented by the black and workers are represented by the gray. And so you can really tell the difference between how the gene is expressed. The ALDH1 gene is significantly expressed in queen honeybees' mandibular glands. Aldehydes are major components of insect sex pheromones, so queens expressing ALDH1 gene can be backed up because queens have hypertrophied reproductive systems compared to workers. As you can see on graph A, queen is represented by black and workers are represented by gray, and you can see that queens are significantly higher. The FAS, fatty acid synthase, gene shows significantly higher expression in workers' mandibular glands. This is likely associated with workers' mandibular glands synthesizing the lipid components of royal jelly. And you can see that on graph D, the difference. The other genes, MCAD and ETFA, graphs B and C, were not significantly different between workers and queens. This is the first study that identified differentially expressed proteins and genes between caste and the honeybee's mandibular glands. Differential hind leg development in caste. The pollen basket or corbicula is on the outer side of the tibia in worker bees. This structure is not present in queen bees. This study by Bontorian, Moda, Luray, Cristano, Suarez, Facioli, Betondo, Barchug, and Simoes reported that the leg structures were formed in pupae in both queens and workers. Using qPCR, the study found the transcription patterns of abdominal A and fat acid binding proteins are involved in the development of leg structure throughout caste differentiation. Conclusion. Queen and worker honeybees have different gene expressions that make them different from each other, even though they're not genetically different. Queen honeybees have an extremely strong reproductive capabilities compared to workers. The expression of fatty acid synthase in workers comes from worker mandibular glands synthesizing royal jelly. The expression of aldehyde genes were significantly higher in queen's mandibular glands, implying that those genes are Cas selective. Abdominal A and fat acid binding protein were found to be involved in leg structure development during Cas differentiation. Putting all of this information together shows us that Cas differentiation is shown in gene expression, and future research can look more at different genes and how they're expressed with queens and workers. And here I have my 5 peer-reviewed research articles as my references.